Hello, everyone. Good morning to those who are on the West Coast. Good afternoon to Human and those who are on the East Coast, and good night to those who are in Europe and also in Asia. I know that there are many people in Europe and in Asia also listening to these seminar series. Welcome uh, to the to this talk today, the seminar series on translational biomedical engineering. I'm very pleased and very excited to have my good friend and colleague, Dr. Stephanie Willerth, today, who will be talking about 3D bioprinting personalized neural tissues for drug screening. So I'll talk about Stephanie in a bit. It takes a little bit of time to talk about her. I mean, I have a personal uh, history with Stephanie since I joined University of Victoria. I'll get to that. But before that, I have a few housekeeping notes. So today's talk is going to be recorded and posted on our YouTube channel, thanks to our speaker who kindly agreed to record it and gave us permission for that. So during the presentation, if you have any questions, please use the ask a question box, which is right at the bottom. If you write your questions in the chat box, we'll probably miss it, and then we may forget to ask that question. So make sure that your question is in the ask a question box. We also have a poll in which you can share your thoughts about the talk, and then also the organization of these e-seminar series, and a few questions that will help us to improve the quality of these e-seminar series. So feel free to participate in this survey, which is in the form of poll. Also, I would like to announce that our next speaker is Professor Alan Eves, the CEO of Stem Cell Technologies, another BC-based company, biotech company. It's a huge and very well-known company in Canada and also in the world. And then he is going to talk about starting a biotech, going over the dark side, or seeing the light. So that's going to be a very exciting talk next week as well. As always, if you need to get some information, most up-to-date information about our e-seminar series, please follow us on Twitter. You can always email me and Human if you have any questions or suggestions, and also Vahid is a coordinator in this e-seminar series. You can always contact him as well if you need to know more about the future talks and then our plans. And if even you know anyone who think could be a good speaker at this e-seminar series, please feel free to nominate them. So we couldn't run this seminar series without the support of our sponsors. TransmedTech has been our continuous sponsor so far. TransmedTech Institute aims to support the development of innovative medical technologies, train the next generation of professionals, and make innovation in life sciences and engineering based on a living lab approach. TransmedTech provides an integrated environment that supports interdisciplinary collaborative processes and co-creation of new medical technologies and interventions to catalyze their development and adoption by users. Our other sponsor is CellLink, who kindly agreed to sponsor Dr. Willer's talk today. So CellLink is producing technologies for 3D bioprinting. Their technologies is their goal and their mission is to create the future of medicine by providing technologies, products, and services to create, understand, and master biology. Their products have been adopted by researchers in more than 60 countries around the world, and 25 pharmaceutical companies in more than 1,800 labs. Their products have been cited in over 150 publications in many application areas, including tissue engineering, cell culturing, in particular 3D cell culturing, and then liquid handling. Feel free to visit their website if you need to get more information about their technology. So with that, now it's time to introduce my friend, my colleague, and then also my mentor, Stephanie Willerth. Stephanie, when I joined UVic, Stephanie was a young professor here. She's still very young, much younger than me. But back then, she was kind of my mentor as well. So she helped me to walk through all these, different aspects of being a professor and a PI. And then she's always there to support not only her students, also other professors and then young faculties. So Dr. Willerth is a full professor in biomedical engineering. She holds a Canada Researcher in biomedical engineering at the University of Victoria. Where she also has a dual appointment in the Department of Mechanical Engineering and Division of Medical Sciences. She also holds an appointment with the School of Biomedical Engineering at the University of British Columbia. She serves as the acting director of the Center for Biomedical Research and the Biomedical Engineering undergraduate program at the University of Victoria. She is also an active member of the steering committee of the BC Regenerative Medicine Initiative and the Stem Cell Network. She also serves as a staff scientist at Creative Destruction Lab. She was the president of the Canadian Biomedical Society. I remember that pretty well, where we both organize, the Canadian Biomaterial Society annual meeting as well. And she is serving a 3-year term as a president-elect in the president and past president from 2017 to 2019 at the Canadian Biomaterial Society. So welcome, Stephanie, to this talk, to this e-seminar series. We are looking forward to hearing your talk on 3D bioprinting of personalized neural tissues for drug screening. The virtual floor is now yours.
Awesome. Well, thank you so much for that wonderful introduction. It's good to see both of you virtually, and even though I can't see the rest of the attendees, it's really exciting to be here. We'll see if I can share my screen. All right. So, let's see if this. Okay. So hopefully everyone can see my slides. As Mohsen said, we're both at the University of Victoria, and this is a bit different from some of the other seminar series I've been participating in COVID. So we'll just all pretend today that you all came to the University of Victoria, which is here on Vancouver Island, north of Seattle and west of Vancouver. And after this is all done, we're going to go out and eat poutine and local fish and Nanaimo bars. And definitely, hopefully, when we get a vaccine and we can travel again, everyone's welcome to come visit us and see the wonderful facilities we have here at the University of Victoria, as well as our growing biomedical engineering program, which Mohsen plays a really important part in. And I think he did a pretty good job covering all the different roles I have. But I wanted to just throw in this slide because I know that there has been, it's a really, really diverse audience, and I know that a lot of people have questions about career path and what different things look like. And as he mentioned, although it gets a bit intimidating when he spells out all my different job titles, I've got a lot going on, but I'm happy to talk about the different roles, especially in the question and answer section, about what it takes to direct either a research center or an undergraduate program, as well as some of the cool things that we have happening in collaboration with the new UBC School of Biomedical Engineering. I also work closely with a lot of different companies, and I also recently spun out a company called Axolotl, which I'll touch on briefly. We do have a pretty exciting biotechnology scene here in British Columbia. Mohsen mentioned Stem Cell Technologies, which is one of the biggest biotechnology companies in Canada, which is based out of Vancouver. And then we also here on island have Starfish Medical, who I've been working with. And some of you may have heard of them because they were awarded the contract. And we've actually, my students are helping them to validate the ventilators to build up Canada's supply of them as well. And I did post in the chat that next Friday we are having our 4th annual Biomedical Engineering Day here at the University of Victoria. It is a virtual event with cash prizes for the best poster, although I think we've closed our posters acceptance for now. But even if you aren't in Victoria, we totally welcome you to come. We have lots of good memories when the event was in person, and we used to have wine with poster sessions, but you can learn a lot about what I hopefully won't be focusing on today, which is some of our efforts here at UVic to deal with the COVID-19 crisis in collaboration with local companies. And also, for those of you who are looking for jobs, hopefully you'll like what you see during my talk and what Dr. Akbari has said about the University of Victoria and opportunities here. But we are looking, as he mentioned, I'm currently the acting director of the biomedical engineering program, but we are looking to hire an external BME director to take over from me at the associate or full professor rank. This job ad is posted, so if you Google for it, you should be able to find it. It's an exciting opportunity. We have about 180 undergrads. They're excellent. We send a lot of them to grad school. We currently have students at Toronto and Calgary and UBC all over. So it's a really great opportunity. And the second position that we'll be advertising shortly is a Canada Research Chair in Assistive Technologies. Here at UVic, we have an organization called CanAssist, which develops adaptive technologies, as you can see here in the slides, for people with disabilities to help them really have a better quality of life. And so again, if you're looking for positions, I strongly recommend that you come here. We're in the process of getting a new building, which they hope to break ground on by the end of the year, and there'll be a whole floor dedicated for biomedical engineering. We also currently have a proposal for a Master's of Engineering in Biomedical Systems at the Ministry, which we hope to have approved by the end of November, and we'll be taking our first intake into that program next fall. So in addition to what I'll be talking about today, there's just a lot of really excitement in this field and in our local community for biomedical engineering and biotechnology. And so Victoria's, all things considered, a pretty great place to be. And also we have pretty low COVID numbers as well. Okay, so with that, I'm going to talk a bit about some of the cool research we've been doing in my lab, and then at the end I'll touch briefly on what my company's done, and I'm happy to open it up to any questions anyone may have. So I think you probably, if you've been coming to these seminars, you've seen people talk about tissue engineering, which combines the principles of biology and engineering to develop functional substitutes for diseased and damaged tissue. And as Mohsen says, I've been at UVic for a while, and this is one of the slides I've been using since I was a brand new professor. And so the way I used to do this slide was, there are things like artificial bladders made out of the Wake Forest Regenerative Medicine Institute that are being transplanted into people. There's the famous Vacanti ear that grew on a mouse. And then I would talk about these engineered artificial tracheas that were being implanted into people. But then back in 2016, it turned out that Paolo Macchiarini, who was implanting these artificial tracheas, it was a very big scandal. It turned out that he had faked several of his credentials and some of the data for this clinical trial. If you're bored during this quarantine and are looking for something to do, since I think they've officially canceled all the fun things you can do for Halloween for this weekend, you can read the Vanity Fair article, which I am a big fan of reading sort of celebrity gossip and magazines like Vanity Fair. So the day we got a tissue engineer into Vanity Fair was quite exciting, although of course we all wish that it hadn't been for the fact that he was running this big scam. Anyway, the area of tissue engineering I focus on is neural tissue engineering. And unlike things like these bladders, that are already going into people. Neural tissue has a bit of ways to go there, although there have been some promising cell therapies that have been transplanted into people, which does give us hope for engineered tissues. And this is one of my favorite slides. It comes from Mark Saltzman's book. And as an engineer, it really appeals to me because it shows sort of the goal, depending on the tissue, whether you're replacing a structure, and that's very common with things like bone or cartilage, replacing metabolism with things like the liver or pancreas. And for those of you who are in BC or in the Canadian Stem Cell Network, you might know that Tim Kiefer over at UBC has some amazing clinical trials going on where they're evaluating the ability of human embryonic stem cell derived islets to treat diabetes. One of the reasons I like working on nerve tissue is because you need to replace both a structure and a function. So you have both the delicate arrangements of these cells in your brain, spinal cord, and peripheral nerve. And then they also have the electrochemical signaling, which allows them to transmit these signals throughout your body. So in particular, why do we need neural tissue engineering? So for those of you who don't work in this space, you probably know someone who's had one of these diseases or disorders. So things like Alzheimer's, Parkinson's, and multiple sclerosis. Current treatments only really alleviate symptoms. They don't represent true cures. And for anyone who's had to deal with someone going through any of these diseases in their most severe form, it's really, really not pleasant. And it's, there's really a clear need to find ways to better treat some of these neurodegenerative disorders associated with aging. Our lab recently has gotten into glioblastoma, which again is one of the deadliest forms of brain cancer, and it's just terrible if someone you know gets it. It's very invasive and recurs, and is pretty much almost a death sentence. On the side of disorders, things like spinal cord injury, which was my first, I would say my first love or my first big area of research. What I focused on as a graduate student in my lab did initially, and you can see Wolf Tetzlaff and Brian Kwan from ICORD, along with Rick Hansen, who did the Man in Motion tour. And these coupling with spinal cord injuries or traumatic brain injuries. And for those of you who know me, I'm a big racing fan. Michael Schumacher, who is possibly the greatest Formula One driver of all time, suffered one of these injuries to his nervous system while skiing. And even with all of his wealth, he made a lot of money when he was a Formula One driver. So even with the best care, the most cutting edge technologies, there's even rumors about him receiving specialized stem cell therapies. He hasn't really improved enough to even be seen in public, or his family doesn't even give regular updates. And so you can see that this is really just an area where we need better ways of finding ways of treating these diseases and disorders. And also shout out to Mohsen, who just recently joined ICORD, which is the British Columbia organization focused on curing spinal cord injury. Both Mohsen and Josh Giles recently joined. So I'm really excited to get us better, to get all of us integrated here in BC. So why is it so hard to find cures for these diseases and disorders? And one of the reasons is the process of drug discovery and development takes a very long time and costs a lot of money. And in particular for neurological drugs, one I've been focused on a lot recently, and I'll show you some of our really cool data on, over 90% of the neurological drugs going to clinic fail. And in Alzheimer's, I think since 2000, they've tested over 240 drugs, with one being approved, and its efficacy is very marginal. So this is an issue, and I actually wrote a commentary about how bioprinting neural tissues using stem cells can be a better tool for finding drug targets for Alzheimer's disease. And one of the reasons why is our current drug screening methods are pretty limited. Most people use animal models because they are relatively cheap and accessible. Well, not necessarily cheap, but they're accessible for all the grad students working out there. A lot of times if you're making these animal models of neurodegenerative disorders, they're quite labor intensive. And then, as I mentioned, they really lack predictive capacity in terms of getting drugs that are successful in an animal model and translating that to people. Although they have gotten more complex in recent years, trying to account for different things, such as gender and sex, and the age of your mice, and things like that. You can also use human tissue slices cultured in vitro. They have a better predictive ability, but they're limited supplies. And just in a callback, 2 days when I used to be able to give these talks in person, and I used to do a lot of community outreach, I used to ask if people would donate their tissues, usually getting the answer no. But then at one of my public Café Scientifique, someone was actually very interested in donating his own tissue for analysis. It was kind of an awkward conversation, but anyway. So what our lab does is we actually use human-induced pluripotent stem cells to engineer our neural tissue. And so this comes from Takahashi and Yamanaka's discovery back in 2006, which I actually saw presented in Toronto at the International Society for Stem Cell Research meeting. And it just blew my mind. So they take an adult cell and they treat them with these reprogramming factors. And they go back to behaving like an embryonic stem cell, which both embryonic stem cells and induced pluripotent stem cells can form pretty much any cell type found in your body, assuming you know the proper cues. And so this was really exciting for me because this meant that we could actually all have our own personalized embryonic stem cell lines, as opposed to embryonic stem cells, which come from an embryo. And most of us, there was one embryo, and that's the embryo that gave rise to us. So we weren't going to have those. But there are actually lots of different induced pluripotent stem cell lines. I'll actually talk about some of our patient-specific work later on in this talk. And again, if you're bored as everything's getting locked down again, I did write an entire book on engineering neural tissue from stem cells, which you can read if you're bored and want to learn more about this topic. So what cells do we actually want to make from our iPSCs? In general, you want to make different types of neurons. They're the excitable cells that transmit information throughout your body using electrochemical signaling. You can have different types of neurons depending on whether they sense or transmit information. They're oligodendrocytes, which essentially act as insulators, and they wrap the smilin around the axon, as you can see in this nice picture here. There are also astrocytes, which help provide nutrients to your neurons and oligodendrocytes. They also help make up the blood-brain barrier, which is the protective layer that surrounds your brain and spinal cord. And while I'm not going to touch on it today, there's some really nice work being done by Cheryl Wellington's group at UBC and Ethan Lippman's group down at Vanderbilt, where they've actually been able to develop models of this blood-brain barrier, because one of the big issues with drugs, getting them to the central nervous system, is getting them to actually cross this blood-brain barrier. Other things, which I won't touch on today, but are also important, are the presence of microglia, or the immune cells of the brain. And also in the brain and spinal cord, there are blood vessels. And so a big challenge, again, that I'm not going to get into much detail today, I'll probably talk on it a bit more next week when I'm at the Pirellis seminar, is how to actually get both vasculature next to healthy brain tissue. And Pirellis has made some really interesting technologies for generating vasculature around essentially organoids. So one of the ways we get these human-induced pluripotent stem cells that can become any cell type found in your body into neural cells is using small molecules. And so these work in a manner similar to growth factors, but tend to be less expensive. And for those of you who work with biomaterials and encapsulation, they're less sensitive to the conditions if you're actually trying to encapsulate them for controlled release applications. As many of you who've worked with growth factors know, these proteins are really sensitive to temperature and pH in terms of function, and they also tend to be pretty expensive. We work with three main small molecules in our group. The first one is retinoic acid, which has been known since 1996 to promote the differentiation of embryonic stem cells into neural lineages, and in particular neurons. It's a vitamin A derivative. It plays an important role in establishing polarity by activating the Hox genes, and especially it's important for forming the hindbrain and the neural tube. Puremorphamine, as you can see here, is a small molecule with a lot of rings, and this is a small molecule agonist against the protein smoothen, which in turn activates the pathway, the Sonic Hedgehog pathway, which yes was named after the video game character. And yes, I believe is now a animated movie that you can also watch in case you don't want to do more science on the weekend. And our group showed that it really does efficiently promote the neuronal differentiation of iPSCs. And in particular, it specifies the neurons that we generate into motor neurons, which are the type of neurons found in your spinal cord. So you can sort of see that, and I'll talk a bit on this as well in the context of engineering spinal cord tissues in a little bit. The third small molecule we use a lot is glucosterone, and this is a naturally occurring plant steroid that's usually used as an anti-cancer drug. And a scientific reports paper by Gonzalez et al. They screened, I think, over 1,000 drugs to see what would efficiently make dopaminergic neurons from human embryonic stem cells. And they found that it was really effective at making this type of neuron. And then Megan Robinson, who's was a former undergrad with me, then my lab manager, and now she currently manages Ryan Flanagan's group over at UBC, where they actually are engineering testy organoids from human-induced pluripotent stem cells. So she was the first author on this paper where we actually showed you could use these molecules to make functional dopaminergic neurons in 2D and 3D from human-induced pluripotent stem cells. Okay, so we've got all of our drugs. As most mentioned, and as Human knows, because we've all been involved with the Canadian Biomaterials Society, what do we want to do with drugs? We want to deliver them in a controlled fashion from biomaterials. So we incorporated these drugs into microspheres, which you can see here are small spherical particles fabricated from biocompatible polymers. And in our case, we used polycaprolactone, given the fact that it's low cost and it's long lasting, and for neural tissue, you want extended drug release, so that's why we chose it. And then we published a series of papers showing we could encapsulate these 3 different drugs into our microspheres and then use them as a tool to engineer neural tissues from human-induced pluripotent stem cells. And so just to briefly cover some really nice work done by my former master's student, Andrew Agnew, who now manages Cheryl Wellington's lab, who I mentioned earlier, doing blood-brain barrier things. He was able to show for the first time we could get controlled delivery of glucosterone inside of our microspheres. And when we incorporated it into our polycaprolactone microspheres, we got extended release over 45 days. At the time, we weren't into bioprinting yet, and so we were actually incorporating these into aggregates of stem cells to promote neural tissue. And so we thought by incorporating these microspheres throughout the aggregate of stem cells, you would get more even differentiation and more mature differentiation of the resulting neural tissue. And here's one of the pictures. Hopefully you can see here is the aggregate that we had incorporated the microspheres into. And then we got all these beautiful long processes that extended out when we had combined these microspheres with our hiPSCs. And in this case, we were trying to produce brain tissues. And Andrew went ahead and quantified the fraction of cells in these tissues that were becoming beta-tubulin 3 positive neurons. And he found that our microspheres more effectively differentiated our tissues into having a high percentage of neurons compared to other controls, including when we added glucosterone in soluble form directly to the media. And so it was suggesting these were a really effective tool for promoting differentiation. And so if you read the paper, or you don't have to, which is why I have a conclusion slide, these glucosterone-releasing microspheres really enhanced hiPSC differentiation into neurons in comparison to both positive and negative controls. And then we also had a live cell imager, so we were able to show that they enhanced the neurite length and branching after day 12 in comparison to negative controls as well. And interestingly enough, our lab currently has on order from CellInc, thanks for sponsoring CellInc, one of their cell site live imaging systems. It's currently, as many people are, it's currently not being allowed in the country because it currently lives in Boston. But hopefully when there's less restrictions on the border, they'll be able to come up and install it. And we're very excited to beta test it and work with them to develop some of these tools to analyze our engineered tissues. Laura de la Vega, who was my superstar PhD student who defended a year ago, and also one of the co-founders of Axolotl, she did similar work, but she was looking at controlled delivery of pure morphamine. And again, she showed for the first time that we could actually deliver this in a controlled fashion, and this work came out in Advanced Biosystems. Again, we were very focused on making small microspheres to ensure they were incorporated properly into our engineered tissues. And this was the first time anyone was able to demonstrate controlled release. So if anyone in the audience is interested in delivering pure morphamine, either for engineering neural tissue, some people use it for skeletal muscle as well. We'll be happy to talk with you about how to measure the release using HPLC, as we did a lot of methods troubleshooting here. And so Laura actually used a combination of retinoic acid releasing microspheres and pure morphamine releasing microspheres to engineer her neural tissues, which, as you can see here, looked really nice, gorgeous, and mature as they extended out, really almost looking like a sun extending sun rays out as well. And this came out in Advanced Biosystems back in 2018. And although I'm not going to show it, she did lots of lovely work on this, showing how these combinations of microspheres really made essentially many spinal cord tissues that express the motor neuron marker chat. And this was again in comparison to positive and negative controls. Interestingly enough, we actually grew these on microelectrode arrays, and they did have spontaneous electrical signaling and activity after 64 days of culture as well. Okay, so to get down to the topic at hand, 3D bioprinting. One of the things you might have noticed with me talking about some of our foundational work that led up to us getting into bioprinting is that when you engineer neural tissues from pluripotent stem cells, it takes a number of weeks and a significant amount of labor. And I'm sure any of you who've met Laura, Ruchi, Andrew, Nima, any of my graduate students will tell you about how long cell culture takes. Megan, for sure. Tara, for sure. And these neural aggregates can extend out in all directions, as you saw with some of those things like I just showed you with Laura's work. Over here is just a picture of an organoid. These are really cool technologies where you harness the intrinsic power of stem cells and you put them under certain conditions, including shaking them, and you treat them with a bunch of different growth factors, and then you get a structure that resembles a mini brain. However, you currently only get what really looks like a nice mini brain 1 out of 4 times, which is why pharmaceutical companies have been slow to adopt them. There are like a number of groups out there working on this technology, including Ethan Lippman's group, a lot of engineers working to make little mini bioreactors to make this process more standardized. And this technique was actually pioneered by Madeline Lancaster, who currently has her own group over in Europe. And she just released, I think, some really nice work with some of her organoid technology and actually getting, I think, cerebral spinal fluid in her organoids and stuff. Anyway, definitely check it out. Lots of really cool science coming out in terms of organoids as well. But essentially what we've heard from pharmaceutical companies is what they really want is reproducibility and high throughput. And that's where 3D bioprinting comes in. So 3D bioprinting, similar to traditional 3D printing, which since I'm at home, I'll just show you a traditional 3D printed axolotl, like the name of my company, done by one of my students, where you have a heated up plastic filament and then it gets printed in a pattern after being extruded from a nozzle. Whereas in 3D bioprinting, you still start the same with the CAD file, but you're instead using specialized bioinks laden with cells to be printed into your pattern to generate a tissue construct. And then you can treat that with your media and growth factors, and then you can mature these into things that resemble either brain tissue or spinal cord tissue or other tissues if you aren't working in the area of neural. And then today I'll be mainly focusing on using them for drug testing and studying fundamental biology. But I do know Aspect Biosystems recently got a grant with Tim Kiefer where they're actually going to try and bioprint therapeutic tissues that would go into humans. And if you're interested in learning more about 3D printing healthy and disease models of brain tissue, Conrad and Simon, who are two of the co-founders of Aspect, and I actually wrote a current opinion paper on this as well that came out. So just in general, not just when bioprinting brains, but there's lots of considerations when bioprinting. How are you going to print? A lot of people have built bioprinters at home. I'll talk about the Aspect RX1 system today. We also have from CellInk a CellInk Incredible. We just recently got a BioX and a LumenX, and we also have an 11V1 here at UVic. So it's pretty exciting. Got a lot of different toys to play with and to choose from. The choice of cell types. Most tissues have multiple cell types, and I'll talk about that. Primary versus stem cell-derived cells. Oftentimes, primary cells are a bit more stable when you're bioprinting. The choice of materials for your bioink. The easiest materials to print aren't necessarily the best for cells. And I'll talk about that in a bit. Your tissue structure. It's still really hard to predict what type of structure will print when you're doing your 3D printing, and what factors to incorporate into your tissues. And that's where you'll see the microspheres again. For those of you who aren't as familiar with bioprinting, well, our group has a couple of reviews out on there. So if you're interested, you can check both of those out. One's on neural tissues, and the other's actually on stem cell-derived tissues in general that we published with our collaborator, Banana Jodar. It was actually, I think, one of the most downloaded papers at Cellular and Molecular Bioengineering for that year. But 4 of the main methods for doing bioprinting include inkjet printing, where you have either thermal or acoustic forces that are extruding your droplets of bioink containing cells. You can also use lasers to pulse and essentially deposit your droplets on the substrate you're trying to collect or engineer tissue on. We work with a subset of extrusion printing that relies on microfluidics. And you can either extrude using air pressure or direct force your bioink containing cells. And I'll show you some of our microfluidic printheads that Aspect's made in the next slide. And one of the last techniques is cell electrospinning, where some of you might be familiar with traditional electrospinning, where you have a polymer solution. You apply a really high voltage field to, and then you pull out these thin streams of either polymer or in this case, polymer and cells. And then depending on the method of collection, you can get different patterns and shapes. So one of our great collaborators, who we've really have been together for a while now, I think since 2016, has been Aspect Biosystems, which is a bioprinting company that spun out of the University of British Columbia. Tamer Mohammed's the CEO. Conrad Wallace was the professor who sort of oversaw that. Great team. We love working with them. And we've used their technology a lot in our lab. And so what's been interesting about their technology is they use these microfluidic printheads, which you can see here. This is one of their older ones that has 4 channels. It allows you to switch between multiple materials when you're printing your tissues, you can see here for one of their demonstrations. And they actually published a paper this year showing how these smooth muscle tissues they make actually contract when using their 3D printing technology. And they've actually got a couple new printheads, including a coaxial one, and I believe a 6-channel one, so you can have up to 4 materials. What we were really interested in is it's a very sophisticated bioprinter, and their flow focusing method allows us to, when you print stem cells, expose them to less shear forces. And if any of you were attending Till and McCullough yesterday, Eric Jervis actually brought this up, that especially pluripotent stem cells are very sensitive to shear. And so at the time we started working with this technology, no groups had really published high levels of viability when printing stem cells. So usually they would print neural stem cells and about 50% of them would die. And so this is why this technology was of interest to us, was that being able to control this and the shear forces meant that we could get a higher level of viability. And just to, again, I know this is a pretty broad audience, so I figured I would touch on sort of the collaboration that I have with Aspects. I think it's been pretty unique and pretty successful. So in 2016, Tamer called me up, and we actually were talking about bioinks, because at that time they were working with mainly their primary alginate-based bioink, and they wanted us to develop some inks for them. So we obtained an Engage grant in 2016. We then were very lucky and got a commercialization impact grant from the Stem Cell Network, which enabled us to bring one of their printers onto campus. Since then, we've been able to leverage that funding to get Innovate BC Ignite funding. We got funding from INSERC, their Idea to Innovation grant, and most recently a Michael Smith Foundation for Health Research in collaboration with the Pacific Parkinson's Research Institute, Idea to Innovation grant to commercialize our neurobioink. Laura, who I mentioned before, she actually did a MyTax internship with Aspect, and that's definitely something, if you're here in Canada, I would highly recommend, is participating in the MyTax program, especially if you aren't sure if you want to be a professor or stay in academia. At least you can see sort of both sides and sort of the differences in culture and expectations between the two settings. As I already mentioned, we've published together multiple papers, and so that's been one of the nice things about our collaboration. Aspect's very happy for us to publish. They haven't really stood in our way of publishing. But yeah, we have 2 papers. We're both co-authors on them. And also, we recently received the 2020 REACH Award for Excellence in Research Partnerships. So this is one of the University of Victoria university-wide research awards. So we won one of the top research awards here at UVic. And this is Conrad, the founder of Aspect, and me when we won our Innovate Award. And they did give us a giant check, which I will just say I was very happy and excited that we did get a giant check. It was a bit disappointing because the giant check is actually erasable, so they reuse them. Otherwise, I would have tried to steal it. But for those of you who remember the Publishers Clearinghouse Lottery, yes, it is quite satisfying to get a giant check. And if anybody's from a funding agency out there, I think that you should give out giant checks whenever people get giant grants, because it's fun. And hopefully someday we'll have award ceremonies in person again. And as I sort of alluded to in the last slide, one of the papers we did collaborate on was a paper where we developed a printable version of the protein fibrin. So for those of you who haven't worked with fibrin, when you cut yourself in a blood clot forms, that's fibrin and fibrinogen at work. So fibrinogen is the polymer that gets cleaved by thrombin into fibrin monomers that assemble into the scaffolds that make up your blood clots. And we've been working with this material. I've been working with this material since 2003. It's wonderful. It's biocompatible. Cells love it. Those of you who work in the bioink fields know it takes a bit of work to get your materials to be printable. And so thanks to Emily and Lila Abelseth, a team of sisters who helped do this and conduct this work. So when we first had our bioink, as I mentioned previous to this point, when you're printing stem cells, neural stem cells, about only 50% would survive. So we printed some very simple constructs to start with, mainly cylinders, which was Laura's idea of trying to sort of mimic the spinal cords, and then the rings, because Aspect really liked printing rings. And so that was, I think, an easy file for us to print. And we were quite pleased with the results. Neural progenitors really enjoyed our ink and were able to grow, proliferate, and mature into neural tissues inside of them. And so I'll just talk a bit about some of the work that Laura did initially with this lab on the printer work. So in this case, again, we use the Aspect system to make these miniature spinal cord constructs. They were 1 centimeter in diameter, and we printed 7 layers of fibers. The fibers had an average diameter of 160 microns. And this work came out in applied science, and what's really nice about that is it's open access, so anyone, anywhere can read this paper. This was building upon Laura's work. So she, as I mentioned, did all that work characterizing pure morphamine. So she was curious, in this case, before, this is just using small molecules, no microspheres yet, but she was curious the effect of treating these different bioprinted neural tissues with different small molecule combinations. And as you can see here, in addition to pure morphamine, which I talked about in retinoic acid, both alone and in combination, there was also this cocktail of 3 small molecules, CHIR, which is a Wnt activator and a very selective inhibitor of GSK3, SB, which is a TGF-beta receptor antagonist, and LND, which is a TGF-beta SMAD inhibitor. So these molecules have been shown to really jumpstart the maturation of your tissues. So they've been shown to really help differentiate these tissues. So that's why those are included. And you can sort of see the different conditions. The negative control was just these small molecules. And then you had the group with pure morphamine in addition to those, retinoic acid in addition, and then a group with just pure morphamine and retinoic acid, and then the other group with all 5 of them. And so here are just some phase contrast images. Again, this was very early days, right when we first got our printer that was, I believe, installed on Valentine's Day of 2018. Very romantic. And we've been waiting a long time to get the printer. And so we were taking phase contrast photos. And I don't know if everyone in the audience at home in their living rooms can see, but the neural progenitors did really like our ink, and they did multiply over time in these phase contrast images. Laura also did viability. And so interestingly enough, we saw much higher rates of viability than had been previously observed when bioprinting, both initially after printing and after 7 days of culture, where all the viabilities were up over 85%. And no differences were observed between the different groups. So the small molecule treatments weren't having an impact on viability. Laura went ahead and stained them, and I believe there's also flow cytometry data in the published paper. But essentially, we saw that these tissues express the neuronal marker beta-tubulin-3, gliofibrillary excitic protein, which is associated with astrocytes, and also the chat motor neuron marker, which I previously mentioned. And as you can see here, there's sort of different intensities depending on the treatment group as well. And as I mentioned, Laura went ahead and quantified this using flow in the paper as well. And so some of the exciting things from this paper were we could actually print these hiPSC-derived neural tissues and maintain them in a high level of viability. Also at the time, bioprinted constructs hadn't been cultured for 30 days. And so getting them to be cultured for such a long period of time and then seeing mature neuronal markers, that was great. And being able to alter the composition with small molecules was also good because it showed we could sort of tune the composition depending on how, on what kind of tissue and what sort of composition we would want. The other interesting thing, I'll talk a bit more when I get into some of our Alzheimer's work, is that a lot of the human-induced pluripotent neural models are mainly done in 2D, but they often only use one cell type. But a lot of work has come out recently. These diseases like Alzheimer's and Parkinson's, really there's a lot of interplay between both the neurons and astrocytes in terms of disease phenotype. And so it's definitely been of interest to the pharmaceutical industry and academic researchers that we can have these tissues where you do have multiple mature neural cell types present. So more recently, my senior PhD graduate student, Ruchi, she wanted to take all that microsphere work that we did and actually use them as a tool to promote differentiation into brain tissue. So here, she used a similar process. She printed a different pattern than Laura. And if you read my review paper, or I can just spoil the ending for you, a lot of the structures that people have printed for neural tissue engineering don't even come close to replicating anything you would find in vivo. A lot of them are cross-hatched patterns, which shows that you can actually 3D print a structure, and it's very easy for measuring the size, shape, and reproducibility of your fibers, but you aren't going to really have a cross-hatch anywhere in your body. There was some really nice work from the McAlpine group at Minnesota where they did do some very nice work patterning some detailed spinal cords that came out in advanced functional materials. But Ruchi decided to print essentially half domes as a first sort of pass at trying to get a shape that was more similar to a brain. Again, as I mentioned, it's kind of challenging to print more complex structures, but she was able to do it after playing with some different parameters. And in this case, she was bioprinting these half domes containing both our neural progenitor cells, along with Google sterone releasing microspheres. And she found in this paper, which came out in Frontiers back in January, that we did get differentiation into these different cell types. And in particular, she was trying to make, as I said, brain tissue and dopaminergic neurons, which express the marker TH. And so she was able to show that with Google sterone that we did get TH positive neurons inside of our little miniature brain tissues. And so what she found was that these microsphere-laden bioinks actually promoted more mature neural differentiation of our neural progenitors than when we just added soluble drug to the media, probably because they're distributed throughout the tissue. Interestingly, they also increase cell survival. We have some hypotheses that potentially the microspheres are having a physical effect on the cells and potentially stimulating them to reproduce more in that sense. She's currently testing different combinations and concentrations of all three types of microspheres to determine their effect on differentiation. And yeah, we are also testing the effect of our microspheres on the rheological properties of our bioink. And she got some really nice data that if you attended the BC RegMed Symposium, she showed on her poster there. And just to give you a taste of where this work's going in the future, we collaborate with Dr. Håkan Nygaard, who runs the Alzheimer's clinic over at UBC, but he also runs an active research lab, and we co-supervise a couple of students together, including Chris Lee, who started out at UVic, and now he's doing his master's over on the mainland, but we have this very active collaboration. He actually has taken, he's derived cell lines from the patients he sees in his Alzheimer's clinic and has characterized them. And so what's really cool is that we've actually been bioprinting neural tissues from these patient-derived hiPSCs. And hopefully, once we get the characterization finished, we can potentially use them to evaluate off-label drug effects to see if we can reverse Alzheimer's. So here's just some pictures of one of the hiPSC lines derived from one of his patients. Essentially, you do a blood draw, and you send it. Francis Lin runs a core facility over there. They do the reprogramming for you. They just recently sent us a Parkinson's line as well. And so Håkan's group got some of these lines from his patients and they differentiate them into cortical neurons. So A is showing the undifferentiated lines and B is showing the cortical neurons after differentiation. And here's just showing that they could produce both mature neurons from their patients along with the astrocyte marker. And interestingly enough, with these cell lines, especially with things like Alzheimer's, where you know what the mutation is that causes Alzheimer's, you can actually go back in and CRISPR and use gene editing to correct them. And so what's very interesting about these cell lines is when you actually culture the hiPSCs in a dish and you turn them into neural tissues, the neural tissues, just like the patients, get more Alzheimer-y over time. And so one of the things you see with Alzheimer's patients is the tau tangles and things and the amyloid beta proteins. And so again, here we can actually measure these in a dish And the Alzheimer's patient tissues show increases in these that are different than just the control cell lines. And so this gives us a way to essentially monitor our Alzheimer's symptoms in a dish. And so while this was done with 2D culture, I've been very excited about work that John Walter Schopke has been doing in my lab. And he actually just presented a poster on this work yesterday at Tillum McCullough. And this is actually one of the reasons I like to show these photos, is this was like the very last experiment that was finished when our lab shut down due to COVID. And we got one exemption for him to essentially finish out taking these Alzheimer's patient tissues to day 45. And then he had to fix them and put them in the fridge for 3 months until the lab reopened. But even with the phase contrast images, you can see that we were starting to get neural projections inside of these tissues. And so he really has this lovely 3D bioprinted Alzheimer's tissue model. Cut to, I guess we're now in October. We were able to get back into lab in July, and he was able to stain these for the neuronal marker TUJ1, which is shown here in green, and blue is DAPI. So we get a really nice distribution of our cells throughout, and then we actually do see these really nice neurite extensions in our mature neural tissues. And he's also currently analyzing all these supernatants for the tau and amyloid beta protein to see if we've replicated in our Alzheimer's tissues what we were seeing in 2D culture. Okay, so that's some of the work, and I think it's really showing the potential of using human-induced pluripotent stem cells as a tool for drug screening and personalized medicine. And now I'm going to just briefly go through a really awesome example of how we've actually been able to use a 3D bioprinted tissue to evaluate a novel drug. And I already mentioned in my background about glioblastoma, which is the most deadly primary central nervous system type of tumor. As its name suggests, it originates in the glial cells in the brain, and it has a very short survival time of 32 to 36 weeks. It's a terrible disease, very deadly, very aggressive, and of course, because it affects cognitive function, just very devastating for the families of those who have to live with their loved ones going through it. So obviously, there was a huge need to develop better treatments. So John, who I mentioned earlier, who's now I co-supervise with HACON as a master's student, as an undergrad, he was really interested and keen on bringing us into this cancer world. And so he had drove this project all by himself and went and acquired us human U87 glioblastoma cells. He bioprinted them into rings, and then similar to what Laura did for our neuroprogenitors, the glioblastoma cells really loved our bioink. And this work came out in Materials Today Chemistry last year. And being an overachiever, he went ahead and characterized it. And it turns out that when you put these terrible glioblastoma tumors in a 3D environment, they behave exactly like they do in the brain. They are very invasive. They spontaneously form more spheroids and tumors. And here's just some phase contrast images of these tumors. And then here's some scanning electron microscopy images of these tumors. But essentially, they're even worse in 3D than they are when you culture them in 2D. And he actually showed in this paper, I'm not going to get into too much depth, like we had previously found a treatment that worked very well at turning cancer cells into neurons in 2D, but then when he actually tried it in 3D, it didn't work. And it's partially due to how the tumors form and how tightly structured they are. In this case, he found that the printed cells, as I already alluded to, expressed increased CD133, which is a glioblastoma stem cell marker, and DCX, which again is highly upregulated in invasive glioblastoma. So letting these cells loose in a 3D printed environment really enhanced their tumorigenicity, or essentially I think was better replicating what happens when you have these tumor cells inside of the 3D environment in the body. So last August, I had an amazing student come to me from the Netherlands, Imke Smits, and she was very excited to bioprint, and we began collaborating with a company called Zonula, who has a bunch of different antagonists, which they had tested in other cancer lines, but not glioblastoma. And so these all target the adhesion molecule in cadherin. And so here we can see the compound they sent us, compound 15, which is one of their incadherin agonists. And we actually have another project currently ongoing with them. And Zonula is based out of Montreal, in case anybody wants to get in touch with them. So for those of you who aren't as familiar with incadherin, it's a cell adhesion molecule that's commonly overexpressed in tumors. It's associated with the epithelial mesenchymal transition, and if you see high expression, it means you're going to have a poor outcome in cancer patient cohorts. And again, expression of incadherin is strongly associated with the glioma grade. So the idea is this is potentially what is helping these tumor cells stick together. If you can disrupt it, you can disrupt the tumor and eliminate cancer, is the working hypothesis. So MCA, being a rock star, went ahead and bioprinted cancer constructs using Chris's procedure that we had published in Materials Today Chemistry. And here you can see, this is just live dead stain. So we had the control ones. We diluted this compound in DMSO, so she had a DMSO control. And then she treated them with different doses of this compound. And you can see the higher the dose, the more toxic it was to these glioblastoma cells. So this was quite promising that when you treat them in the bioprinted constructs with this drug, that we were actually seeing some cell death. And being such a rock star, MCA actually printed a very complex model of the neural tissues trying to replicate what you would see in a brain cancer situation. So she printed healthy human astrocytes alongside these glioblastoma cells. And for those of you that are wondering, we include phenol red when we bioprint our constructs because the fibrin is normally pretty clear. And so by including phenol red, it helps us visualize where in the microfluidic device the fibrin is, and it helps visualize clogging, and also helps you look at the structures as well. And this is just an image showing our construct as well. And so what IMCA did was she actually allowed these co-culture models for all the groups. So no treatment was applied for the first 16 days. And so as you can see here, all the groups got these very large tumor formation. I also want to point out that she used GFP labeled tumor cells and the astrocytes were unlabeled. That'll be very important in a second. And so what she found was, so at day 16, remember, we haven't treated with anything at this point, so we just have a large number of very green, very bright tumors. She then treated with these different concentrations of this drug, the 5 micromolar, 10 micromolar, and 25 micromolar. And by day 22, even just visually, you can see that there was a large decrease in the amount of green tumor cells present in the 2 higher doses. And even by day 30, there was a huge decrease in the amount of GFP present in these cultures. So this was suggesting that it was actually selectively killing off the tumor cells while leaving behind the healthy astrocytes. And she goes into more detail in her paper, which was published in Biochemical and Biophysical Research Communications. And that's, you can see here the citation. Here's, she quantified all the fluorescence. So you can see at day 22, there was a significant decrease in the fluorescence compared to the controls. And then the same at day 30 with all groups being different compared to the controls. So this really helped us show and validate that this compound, this novel incadherin antagonist, causes glioblastoma death. And if you read the paper, and we have a ton of supplementary data, she really elegantly shows why we needed the bioprinting and the co-culture model to show this in comparison to traditional 2D culture models as well. And also she did some work where she actually casted these cells into fibrin gels, and it really showed the advantages of using these 3D bioprinted constructs. So conclusions from this. We successfully printed a novel co-culture model where we have both glioblastoma cells and healthy astrocytes. And we hope to build on this in the future to, again, build more complex models of tissues. If anyone out there has primary patient glioblastoma cells and is looking for a collaborator, we would love to collaborate. We also identified a novel compound that was toxic to glioblastoma and could shrink their tumor spheroids, which I think is a much more clinically relevant model than traditional 2D cell culture. And as I mentioned, IMCA sort of validated that with a lot of her supplementary data. And yeah, as I mentioned, would love to try this on patient glioblastoma cell lines. Also, I know Orest, who is the CEO of Zonula, he also is looking to test this in animal models. So if anybody does animal cancer models, I'd also suggest reaching out to him as well. Okay, and then just with my final few minutes, I just wanted to spend a little bit of time on Axolotl and starting a company. So the week of the pandemic, as you know, a lot of people, the pandemic made us all act very weird and do things we didn't do. A lot of people played Animal Crossing or learned how to paint and got hobbies. What we did instead, Laura, Lila, and I decided to incorporate a spin-off company to sell our novel bioinks, the fibrin-based bioinks, both the base ink and with microspheres. So we created our company, Axolotl Biosciences, whose goal is to enable 3D printing of humanized tissue. And our goal was to, of course, manufacture and sell bioink as soon as possible, which was a bit difficult with our lab shutdown. But it's actually been working out surprisingly well. So I already mentioned Laura and Lila, who obviously were key, instrumental in doing a lot of this bioprinting work, and so they're my co-founders. I'm currently completing the W Venture Accelerator, which is part of the digital supercluster here in British Columbia. And Axolotl was recently accepted into the Mars Accelerator over in Toronto, so we're quite excited about that. We also recently were accepted into Life Sciences BC Investor Readiness Program. So it's all been really quick. It's all been happening since March. We're at 5 full-time staff in summer, including Josh, who you can see here, who's still working for the company, and Callie, who is our full-time production manager. We recently brought on Brett, who is a full-time business development manager, and Emily Abelseth, who's our quality analyst. And we have a number of formal and informal advisors. But we've been doing a lot of work to get our ink out there and being tested by people, as well as doing market research, signing licensing agreements. And it's just been a really interesting experience that's a bit different from sort of your typical academic activities. Essentially, our vision as a company is to deliver high-quality products to enable the 3D bioprinting of humanized tissue models for advancing healthcare. We want to provide turnkey reagents, like our bioinks, tissue models, and consulting services to help advance tissue engineering and regenerative medicine, and really to try and get better cures for not just neurodegenerative diseases, but all of these diseases by collaborating with different groups to make these humanized tissue models. And our values are consistency, innovation, collaboration, integrity, and leadership. We're really excited. So even despite all the COVID-related setbacks, we currently have our ink in a number of different countries. And it's currently in beta, but CellInk, again, shout out CellInk, thanks for sponsoring this. We currently have our ink at CellInk being tested. We also have it with Printer, a Finnish company. Benata Jodar, who's our collaborator I mentioned before, we co-authored that stem cell review together. She has been testing it in the context of cardiomyocytes down at the University of Texas, El Paso. We have a number of groups at UBC testing our ink, in addition to my own lab. And we've also been in talks with several other groups as well to become beta testers for our group. Hopefully we'll be finished licensing our technology by the end of the month, so we'll be able to sell, and then anyone who'd like to buy our ink can buy it and try it out. Our base fibrin ink has been really well. As I mentioned, cardiomyocytes and a lot of other human induced pluripotent derived stem cell lineages it successfully supports. We've been receiving a lot of positive feedback on that. And so if you're interested in learning more about Axolotl, you can keep up with us on social media. We have a Twitter account, a LinkedIn account, and I think there's some pretty pictures on Instagram as well. And so with that, all the work done here that I've been presenting today was done by the Willerth Laboratory, especially Laura, Ruchi, Lila, Imka, Chris, all of our funding agencies. We couldn't do it without them. Special shout outs to NSF in the United States and FICEPI in Brazil. Our international collaborations are very important to us, and it has been very disappointing not being able to bring in international collaborators like we used to, but hopefully it will change. And if you're interested, follow me on Twitter. So with that, I'm going to stop sharing, and then I can do a question and answer.
Thank you so much, Professor Willerth, for your interesting talk and your journey towards neural tissue engineering. So I would love to try your bioink also in cardiac tissue engineering. As you said, it's working with cardiomyocytes.
Yeah, definitely. Get in touch.
Yeah. So before we go to Q&A, you can take a breath, and then I go through a little bit of advertisement here. For our next speaker, we have Dr. Alan Ivas from Stem Cell Technologies. He is the president and CEO of Stem Cell Technologies. He's talking about starting a biotech, going over to the dark side of seeing the light. And then we would also want to thank again our sponsor, Montreal Transmedtech Institute and Cell Inc. Company. By that, I go directly to Q&A box. We got some questions about your technology. The first question from Daisy Hell. Is it a problem that the microspheres for drug release are not the same size and therefore different amount of drug is stored?
I'm really happy to take that. We actually are working with virus group to make them more consistent in size. Interestingly, now that we've moved to the bioink, size is less important. We can have them a bit bigger, start actually incorporating them into the aggregates of stem cells, but we're using microfluidics to make them more consistent for our bioink.
Great.
We have some echo.
Okay, so Mohsen, you go ahead. I bring my headphone. You go ahead with the other questions. I bring my headphone.
Okay, all right. All right. So the next question is okay. So the next question is again by Daisy. So is it necessary to apply action potential to neurocytes to keep them differentiated, or how do you maintain the differentiation and avoid de-differentiation?
Yeah, we culture them in, we have if you read our papers, there's like a number of different media changes we go through, but usually when we get them out to past day 30, we're culturing them in brain fizz, which is specifically designed to keep them active. And we have measured, I don't think we've published it yet for our bioprinted tissues, but we have it, Laura's last paper that we're submitting this month. We still see pretty decent maintenance of the action potentials when we've done some calcium analysis from day 30 to 45, suggesting they stay differentiated. I think once our neurons are differentiated, we don't get de-differentiation, but I do know if you, and that's I think why brain fizz helps maintain that function, but I think with a lot of our tissues, you still have a neural progenitor population that keeps replicating, so that can contribute. So I don't think there's necessarily de-differentiation, but sometimes if you have a lot of the growth factor rich media, you actually will get more neural progenitors in addition to your mature neurons that are there.
Okay, thank you so much. I hope that you can hear me without a problem.
Yeah.
So, again, a question about differentiation of these cells and adding these differentiation factors. They want to know specifically about Nike wants to know specifically about the concentration of these differentiation factors that you use to induce the differentiation, and how do you maintain this the sterility and the viability of the cells throughout the 3D bioprinting process?
Yeah, so we try to replicate based on the concentration, although I think that they have lower dosing. We're essentially trying to replicate what the cells would see. For retinoic acid, I think it's 500 micromolar, and pure morphamine it's 1, or 500 millimolar, and it's 1 micromolar for pure morphamine, but that's sort of the range for those type of drugs for getting differentiation. And then we've scaled it so then it's sort of based on what we know, we have the concentration of release over time, so it's sort of trying to replicate that with our microspheres. For sterility and viability, and I will say that this is one of those things that does not get talked about enough. We actually do have our Aspect system in a hood. We have the sealed containers that we've ordered. So essentially, you can culture your cells inside of. They're like little mini, they go inside your incubator, but they also have their own HEPA filter, so you have their own things. So essentially, you would have all the cells are done sterilely in the hood, and we usually put them in these little sort of carriers because for anyone who was lucky enough to visit before we all got closed down, we have 2 TC rooms. So we have one room where we actually do most of our cell culture, and then we have the other room where we do our bioprinting. So all of them are done in a hood, and then we use these little HEPA filtered incubator boxes to take stuff between the 2 rooms. I do know groups that bioprint just on the bench top and then throw in antibiotics as well. And so that's another thing which people don't really talk about, because I do know, like, to just be very practical, like our hood was very expensive that we put the Aspect system in because it doesn't fit in a standard hood. So they had to special order it. So I know that, like, and getting a BSL2 hood specifically for printers has been an issue. So some people just try to be very clean and print on a bench and then put them in immediately. The new BioX actually does, and this is not a paid advertisement from selling, but the new BioX does actually self-sterilize. So does the BioAssembly bot, which is another way of doing it. But yeah, that's sort of how we do it. Interestingly enough, on the cell viability issue, which might be particular to our bioink, is to get the number of cells we need to print our neural tissues. My students actually culture all the neural progenitors and freeze them down. And so all those viabilities you've seen are reported are actually thawed cells going into our ink. So we're getting over 90% viability, which also includes the thawing process, which was very interesting to me, knowing that, like, you usually are going to lose cells during freeze thaw anyway, that we got that good of recovery. And it might be due to the fibrin sort of protecting them, like, post thaw. Yeah, but those are really good questions.
Thank you so much. Yes, great. The other question is about the issues that you're facing once you're printing your tissues. The most important issues in the process in neural bioprinting. What are those specific issues you got?
Well, I mean, one of the things with bioprinting, I think, is reproducibility, like getting the desired resolution. As I mentioned, the structures haven't necessarily been physiologically relevant that most people are printing. It's been very hard to get really detailed defined structures, although we are trying. Our next goal is to actually start to print the half domes that actually have hippocampal regions, so we can actually put the microspheres in different regions. So that'd be really cool to actually get patterning in there. One of the things that isn't necessarily an issue, but like are things to consider when you bioprint, for example, which is why we use like the GFP labeled cancer cells. I didn't actually show it today, but you can get fluorescently labeled microspheres, because if you put in a pure morphine microsphere or a Google sterile microsphere, both made from PCL, they're going to look the same in your image. So we often put in small quantities of fluorescent microspheres, so we know that we're actually getting the spheres we want where we want them in our 3D structure. And so I think there's a lot of stuff like that that's just important to consider. And then, although I didn't show it today, functionality is really important, and like showing for like Alzheimer's disease phenotypes, for the Parkinson's, we actually get the dopamine secretion and then you get dysregulated dopamine in the patient ones. So.
Thank you so much. Sunita wants to know about the vascularization. Do you try any vascularization in your tissues, or you don't need it at all?
For drug screening, not necessarily with Alzheimer's. We did, as I mentioned, with Pirellis. Pirellis has sent us some baskets, and we've actually printed neural tissue on top of them, because then you could seed in. So Pirellis makes these vasculature structures, and then you can actually seed them back in with like HUVACs to get vasculature. We just recently got a Luminex, which in theory should allow us to make vasculature. Using Jordan Miller's lovely technology. I'm sure everyone saw his science paper where they made the really awesome vasculature. It'll definitely be a challenge. I think we're probably more likely to be blood-brain barriering it, but actually I'm talking about the Pirellis thing next week, so I'm actually curious. So we printed them and they looked okay. I think it was just really hard to image with the actual Pirellis structure in them. So it'll be interesting to see sort of how that progresses. Obviously with COVID, we've slowed down with everyone being in shifts and things, but it's definitely an important question to answer. Not just for neural tissue, but like if you want larger constructs. And then, yeah, I think like I personally see our lab moving, if we move it closer to that, it would probably be more blood-brain barrier. Although I think Ruchi in my group is really like wants to shoot for the stars and, have all the different cells and vasculature too. But yeah, it's definitely an important consideration.
Yeah, thank you so much. I think I have one more question for myself, and then I will pass to Mohsen. So because I'm working also on this field, in this field, so I just want to know what is the process of validation and for this 3D bioprinting process, and afterwards the engineered tissue to get to the market. Is there any is it crystal clear, or is it still on the under a study or under progress?
Yeah, well, I think those sort of two different paths. And actually, right before COVID shut everything down, I was over in Vancouver at a meeting. It was the Canadian and the UK consulate had a joint meeting. I was actually talking to one of the representatives from Health Canada, and they were really interested in how we can incorporate, and this is something I've actually been talking with Tamer at Aspect about, and Sam Wadsworth as well, and you would be a good person to have as well. I've been talking with, so we talked with Health Canada, and Health Canada's like, well, can you tell us like what the benchmark should be for an engineered tissue, but not to go into a person, but to instead like, what would the 3D bioprinted tissue look like that would replace a mouse? And like, what would you want to see? And so I'm thinking, I talked with Sam and Aaron at Aspect about this, and we're hoping to at some point put together a panel to bring in like Health Canada, myself, people like you, to sort of get that, because Health Canada is really interested in moving towards that and bringing in these new technologies. They just need to sort of, what they said is they want to define the sandbox for what it would be. So that's one part. As I mentioned, it's very interesting to me knowing, obviously I know Aspect quite well, and knowing Tim Kiefer quite well and his clinical trials, that Aspect is going to try and bioprint these therapeutic tissues. And I think it's great. I think it's really ambitious. And just intriguingly, from the regulatory standpoint, you have to go through it both as a cell therapy and as a device. Although in their case, for the islets for diabetes, that's actually like standard, even if you have, I don't know if you're familiar with like the ViaCyte, the thing that immunoprotects the cells, like they have to do it anyway in diabetes for both. So I guess it's less of a burden, but that's something I see as sort of a hurdle. Especially in neural, because you have like there's a lot out there for retinal with cell therapies, Parkinson's, and then I think it's just easier. I think it's already hard enough to get something in the nervous system that like bioprinting's a bit away. Also, since it's almost Halloween, man, I didn't do any Halloween themed slides. I'm a bit disappointed. There's actually really concern about how large you can print a brain construct, and if they actually generate thought. And at some point at UVic, I told this story, and then one of the English majors was like, I'm going to write a whole story about like thinking brains in a dish and like how humanity is oppressing them, which, but yeah, so those are just, I think, some of the things to market. We've had a ton of interest in our bioink, so that's been quite promising for us to see.
Thank you so much. Mohsen, please go ahead with your question. I know that you have tons of questions.
Well, thank you very much, Stephanie. As always, great presentations. And I just have one question because we're running out of time. And my question is, so you have this bioink, and then you build it in a lab. How do you keep it sterile when you want to send it out to your customers? So let's say that you want to make these vials of your bioinks. So do you prepare them in a sterile environment and then send it out, or you sterilize them afterwards?
No, we sterilize it first, and we sterilize into the containers, and then we ship it frozen on dry ice to all of our people.
So what is the method of sterilization that you are using?
Filtration.
Filtration. Okay, cool. And that's is there a particular reason you chose filtration over other methods of sterilization?
Yeah, fibrin's a protein. I think we autoclave our crosslinker because you can, but yeah, with the fibrin, it's usually filtration. And I know that now that we're actually commercializing everything, we're relooking, because all this is, taking it from your lab to scale. I know that like Emily's been looking at other ways of potentially doing sterilization. We also did a lot of work actually seeing, because we're currently shipping it as frozen liquid at -80. We also did a lot of work trying to figure out if we could even just like lyophilize it, and then they would reconstitute it to get longer shelf life. But currently it's got like, I think a shelf life of like 3 months before you thaw, and then, you know, a couple weeks and a month once it's been thawed. But yeah, we're still sort of working on all those things. But yeah, it's been interesting, sort of, you know, with the commercial side versus
it is
just in our lab.
Certainly. And then maybe another question now that we're talking about you know, having two hats, and then both of us, you and I, we have two hats, and one thing, like, one is just the professor hat, the other one is the company hat. One challenge that I have faced is that it's very difficult to balance between these two lives, right? And there are, even the pace is different. I mean the company, if you want to be competitive, you have to, you know, be fast. And then in academia, you're mostly, things are a little bit slower, but you're mostly focusing on publishing papers. So how do you balance between these two lives, these two words, I would say?
Yeah, well, hopefully it will get better. Like, as you mentioned, I already have like 4,000 jobs. And then the part we don't talk about is that, like, I also have all this COVID stuff that I just put in a box, like where we made all the face shields and we track COVID and wastewater and we're making ventilators. In this case, well, this is so bad to say, but with COVID, so I used to travel a ton. And so not traveling has actually given me more time. I have a really good team, and honestly, getting a business director of business development has really helped a lot. The other thing was, we, you know, in a perfect world, my plan was we were going to immediately commercialize and immediately start selling bioink, and that of course didn't happen with COVID. So we already had a product that was pretty far along in development and ready to go. The other thing is we had built-in market, so people have already, like, we only started a company because everyone was asking me to sell it, and then I was just kind of like, fine, we'll do this. And then Laura was really interested. And so that sense, like, I think the other thing, being a bit more established and having collaborators has been really good, because like it's helpful when you aren't the sole, like when you're a new PI, it's like it's most important for you to get out your papers and things like that. And then now it's sort of like with collaborations, you sort of still see the papers and stuff. And then we were very lucky to have like a very good year with grant funding. So it was very weird that like the time I finally had like a very financially secure lab, which I'll admit there were definitely years pre-tenure where it was not like that. That was like the weirdest thing ever, is that with COVID, then all of a sudden, you know, I'm sure your labs are the same. Like, you normally spend so much money a month, and then you go to spending nothing. And then I don't know if it's COVID made me more happy to take a risk and start a company. But yeah, a lot changed. So, and so, yeah, I think that the main thing is to just really have passion for your science, and then have passionate people working for you, and then that'll help inspire you. The nice thing with Axolotl was a lot of people really wanted me to start a company. So there's been like a lot of funding and support, both from UVic, from NIC IRAP, from Innovate BC, like, in terms of things where like I wouldn't necessarily be as enthusiastic. The other thing is, now that I'm a full professor and I've published, I guess, 65 papers, just doing something a bit different has been interesting as well. And then the other thing is just like the reception from the community. Like, CellInc was really supportive, like just like everybody wanting to beta test. It's really interesting when you write your business plan, you're like, here's who we're going to compete with. And instead everybody's like, no, we'd rather just like work together and distribute. So that's been great. And I think the BC RegMed community right now is like in a really good space with, you know, Peter Zanstra coming over at the UBC school. And to be frank, I think in Victoria, if we want to stay here, like, it makes sense for us to have, you know, to be with Starfish, you know, to be a biotech hub. We do have relatively cheaper real estate. I'm also, but with the work with Starfish, I think, and this is something, I don't know if it's come up with you, Mohsen, but IRAP really wants Axolotl to move off of UVic and actually for us to have something like a stem cell technologies type manufacturing facility where we're BSL2 off campus. And I think that's something that would really benefit the community. And I think it makes sense with our BME, with our healthcare stuff. You know, there's stuff going on out here on our island as well, in addition to UBC. Oh, the other thing, Mohsen, which I know doesn't help you as much, but I do have amazing staff, and I have amazing staff support. So I have an amazing lab manager, Nadia. Lila Abelseth, who co-founded my company, was actually my administrative assistant, CBR, and like ran my life. And then in summer, Stephen Bradley helped with the COVID things, and then Mimi helps with the undergrad program. So like, if you have a really, really strong staff that you can trust and like you can delegate to, that's very, very important. And that's something I would say for all, even if you're junior faculty, just even if you can find ways to get staff to help support you, even if it's a part-time lab manager, like it like is very helpful if they organize like your things. So
Great. Well, thank you very much. So I have no other questions, but it was there were many good advices and then very good tips actually for balancing your life. Team is as one thing that we have heard a lot during these e-seminar series is that forming a great team is always basically a way to become more successful. And it may take some time to form a team. It may take some time to establish the collaborations, but after sometimes, once you establish them and then once you form your team, things are going to be pretty much easier than they used to be. And you can expand it. So I have no more questions, Human.
Yeah, just before closing, if you look at the chat box, people are really appreciating your
yeah, thanks everyone for coming.
And then, so by that, I think we can close. And just one reminder, next week, Dr. Alan Ivas from Stem Cell Technologies, same time, same place, same day. We will put the flyer online, so just make sure to attend. Thank you so much, Professor Willerth, and thank you so much, Mohsen, and have a great day, everybody.
Good everyone.